Hello, everybody. I wish everything is fine. Finally, we come to the last lecture in session one about utilization of NCBI MSAV in studying COVID-19. This is an example, and you can use the steps we are going to use here for doing any studying in your research. So simply in this example, we can just try to remind you first that there is many coloring scheme for DNA and many coloring scheme for protein and MSAV. So you can use whatever you want based on the purpose of the research, as we said in the last lecture. So in this lecture, actually, we will just pick up some of these sequences. And as we said, this training actually is based on the NCBI training video, but we will just try to do the same thing here with this example. So simply you can send a set of COVID-19 to the MSAV. So we'll first of all, we will just pick up the sequences what we want to make to use for multiple sequence alignment first. So go to the database NCBI virus and try to start with the SARS-CoV-2 data hub. From this data hub, you can select your sequences. After selecting your sequences, we will continue making the multiple sequence alignment and then coloring. But first of all, I would like to remind you, you can do the same thing with if you're doing the BLAST result. So you can do the same thing with if you have a BLAST result also, you can pick sequences and do the alignment and do the same thing. So let's continue. Now, each page from your result from the SARS-CoV-2 data hub containing about 200 sequences. So this 200 sequences, you can pick them or pick some of them based on what you want to do. First, here for this example, we'll just pick the 200 sequence from the first page, for example. And then you can do the second page, which will contain another 200 sequence if you would like to add more sequences on the next page. And then, of course, you can add much more because people can go to up to 500 if you want to make multiple sequence alignment. And then after picking up your sequences, the sequences you would like to compare with, go here in the bottom and at the arrow number 1 and just press here align. So first of all, we will pick our sequences either from BLAST after doing BLAST or from here after this just searching for from this database SARS-CoV-2 data hub. And then after we pick a sequences, we have to press here align. So when you press here align, you will make your multiple sequence alignment. So he will send your sequences to the couple to make the alignment. And then after that, it will be presented, the result will be presented by MSA viewer. So when you get to the point when you can see the MSA viewer and you can see your multiple sequence alignment, just if you would like to search for the differences between the sequences, this will be the default actually result for you. The default result is show differences. The selection for coloring will be show difference as we said before. So in this default, you can see the red lines here. Under the red line here, as you can see in the red circle here, this circle actually circling the differences, the line, these small red lines. This small red lines here is actually denote differences in that sequence. This is the position of differences. And if you go a little bit down, you can see this is exactly where these differences are. So if you go here, you can zoom to the sequence level to see any region if you want. So you can go here and use the zoom tool, plus or minus, or you can just press here at the letter T. Letter T will let you zoom to the sequence level to any region. Any region you can see by just picking it and just press here at T, this letter T. So when you look at it, you can just, of course, as we said before, you can click and drag the zoom into one of these region, any region you would like to see. Here in this one, as you can see here in the red color, this is a place where you can find differences. So this position, there is a lot of differences here. If you would like to see much more information about this difference or about this SNP, single nucleotide morphism, for example, and you would like to know more information about it, you just right click right click on that position like here, so you will get this pop-up menu which will be existed here, as we can show you at the arrow number 2. So the arrow number 2 is the position which is here. I just right click on that position, so I get this information. This information will be about where this sequence came from, the title of the sequence, the alignment position, the sequence position, and many other information about this position exactly. So you can take a look about this, for example, mutation, and talk about it and why it's a little bit not exist in other sequences, but only it exists in that sequence only. Uh, on the other hand, the here in arrow number 1, you can see that this column, this position actually contain always mutation at that position, and this mutation is from C to T, so from cytosine to thymine. So this position is always having this, it's frequently having this mutation. It happens many times here, as you see, and more frequent than that one. So that's why you can see and differentiate between mutations and SNPs in your sequences and make your study. Uh, as we said, it will be always when you open the program, it will come always to as a default to show differences. This will be um, your default choice. So this method highlight differences observed from the master sequence and the alignment. In order to see any correlation with this scheme, you must select a master sequence. So first you have to select a master sequence to see the difference between other sequence to that master sequence. You can adjust the master sequence on the fly and display differences by selecting a new master from the context menu. So usually they use the consensus as a master sequence, but you can, as we said, you can use any of that sequences and use it as a master sequences. So consensus row set by default, they use it as a master sequence because this is the default, this is the consensus sequence here, up here, the one in the top here. And you can, of course, right click on it and get information about it. But if you would like to, you can default, it will the program will set this as the master sequence. But if you would like to choose other sequence for as a master sequence, you can right click on it and just provide it many option. If you just put your right click on the master sequence here, you can download it. You can show this consensus or whatever, any sequence you want. Either here I just press on the on the consensus sequence. Just if you right click on it, you get many options, as you can see. Here in this slide, actually, we just choose another sequence as an anchor. So you can choose any one of these sequences to use it as a master sequence. You just right click on it and you just get pop-up menu, and from this context menu, you will just press on here in the arrow number 2, set this one as an anchor. So you can use this, for example, as an anchor. You can do the same to any of these sequences. So use it as an anchor or as a master, and then compare the other sequences to that one. So it's very easy to do it. And of course, it's based on your research, as we said, which one you would like to use as a master sequence. If you let the default, it will make use the consensus as a master sequence. Okay, you can choose what column to see as we can see here. If you right click on the columns, as here, if you press at arrow number 1, there is here certain columns. If you open this column, you can get many names for columns, so you can choose what column you would like to see. There's many columns here, names. Columns for sequence ID, column for start, end, organism, collection date, country. There's many column. Which column you would like to see in your as a default in your result? So, for example, here in column and arrow number 2, I just choose these two columns, the organism and and country, and I can of course take whatever I want and remove whatever I want. You can also click to choose the column width. I mean, if you just try to increase the width of the column, you just press and this and that number, and you can increase the column width as you can see. All added values are about the consensus. As you can see, you can add here identity mismatch. So this is comparing to the consensus, as we said. If you have a anchor, another anchor, this information will be about that anchor. But the information here, the identity and mismatch, is actually comparing to the, as we said, to the consensus. You can arrange the column result in ascending or descending order, of course. From here, from the count, you can ascending or descending order based on what you want. So arranging your sequences is very easy. You can choose and hide any rows from the table. Of course, if you would like to hide some rows or delete them, this will be also just pick up them, just pick up them, select the sequences that you want to delete, like this first one, second one, third one, and that one here, and then just right click and then just press here at arrow number 2, hide sequence rows. So it will hide the sequence rows for you if you don't like to see it, or even you can remove it if you want. So this is very good thing to do, and also you can also choose and hide any rows. So not just sequence, but any rows you want from here. From if you press down here, as you can see here, if you want to remove the rows that you don't want them to be included in your work, just press here and the rows at arrow number 1 and choose whatever rows you would like to delete or don't include them in your work. Just uncheck this here, this box, small box. Just remove the check from here. It will not be included in your result. The total row has changed. If you, of course, once you did this, the total number of rows will be changed. Right click to show all rows again. If you would like to show them again, just here, just press here, right click on the and this rows, and then show all rows. So in this case, you will show all the rows. So you can hide some rows, or you can show all if you want. So you can, as you said, you can sort also the rows by identity here, by in ascending or descending order, if you would like to do this here. Okay, in this example, for example, we will look for a common polymorphism in the spike protein in this position. If you can see this position, for example, this very good example. And if you compare your DNA sequence, you will find this mutation, CAT, which is the amino acid D, change it to the amino acid G in the spike protein. This position is 614. In this position, the D is changed to G. So, and this is a very common polymorphism. So you can see in your sequences, for example, you would like to see if this mutation, common mutation in the spike protein exists or not, or you have something else. So that's why you need to make a multiple sequence alignment first, and then from here, using MSAV, you can try to spot these differences and spot this mutation if it exists or not in your sequences under study. So this is one of the cases that people use MSAV for. Identify the position of common spike protein polymorphism mutation in different genomes, like here. So I just gathered some genomes and tried to spot this mutation if it exists or not in that position, the position that we know that it's always have a mutation, mutable position. So that's why this position, we just see that it has been changed from G to D here. And of course, you can use any sequences as an anchor, as we said, and the differences in the feature annotation mark the sequence you are viewing. So this is one of the good things, that you can use any of these sequence as an anchor. So you can use, if you have a wild type, you want to compare this wild type to this sequence. And we don't know whatever you want in your study. Maybe you can have a local strain, you want to use it as an anchor, or you have international strain, we would like to use it as an anchor. So what type of virus, for example, or sequence which one you want to use it as an anchor? So this, as we said, is easy to set using the MSAV. Also, you can save your aligned sequence in FASTA format by just click here, download, and then as an arrow number 1, can see that FASTA and FASTA format alignment as a FASTA alignment, and you can open it with other program if you want to just put your alignment in FASTA format. Also, you can use the friendly printer or friendly PDF or SVG. Also, you can have this as a PDF, the result as a PDF. So you can also share this view with customized columns and rows just by sending a link. So you can here, and using what we call link to view. This link to view, which at arrow number 1, you can just press here, link to view. When you press here, you get the link. A link you can send it to your colleague and the people who are working with you in a certain project, for example, to send them what you can see or what you have already done, and they can see what you already what you see already. So that's why it's very useful sometimes to let your colleagues see what you already saw in that research or in that experiment. And of course, as we always say, you can always send your feedback to the program and get more information if you want. So hope you enjoy and get more information from this session, and I hope you enjoy it, and I hope you practice with some question here, and see you in next session. Thank you and goodbye.